Welcome to the intricate world of eukaryotic cells. These microscopic marvels are the fundamental units of life for a vast array of organisms. From the smallest algae to the largest animals, eukaryotic cells form the very essence of their being. In this exploration, we'll journey into their inner workings, uncovering the secrets that make life possible. Imagine a bustling metropolis teeming with activity. Eukaryotic cells are much the same, with specialized compartments working tirelessly to maintain life. These compartments, known as organelles, each have unique roles to play in the grand symphony of the cell. Just as a city relies on infrastructure and organization, so too do eukaryotic cells. Their intricate structure and function are a testament to the elegance and complexity of the natural world. Join us as we delve into this microscopic realm, where we'll uncover the secrets of these remarkable biological machines. Eukaryotic cells are distinguished from their simpler counterparts, prokaryotic cells, by the presence of a true nucleus and other membrane-bound organelles. This fundamental difference has profound implications for their structure and function. The nucleus, often described as the cell's control center, houses the genetic material, DNA. This blueprint of life dictates the cell's activities, ensuring the faithful inheritance of traits from one generation to the next. Beyond the nucleus lies a labyrinth of interconnected compartments, each with a specialized role. These organelles work together seamlessly, ensuring the smooth operation of this complex biological machine. From energy production to waste disposal, every aspect of cellular life is meticulously orchestrated. In the following sections, we'll embark on a journey through this microscopic world, exploring the structure and function of each organelle in detail. Our journey into the eukaryotic cell begins with the cytoskeleton, a dynamic network of protein filaments that crisscrosses the cytoplasm. Like the scaffolding of a building, the cytoskeleton provides structural support, giving the cell its shape and mechanical integrity. But it's far more than just a static framework. The cytoskeleton is a dynamic and adaptable structure, constantly remodeling itself to meet the cell's ever-changing needs. It plays a crucial role in cell movement, both internal and external. Organelles are transported along these cellular highways, ensuring the efficient delivery of molecules and materials throughout the cell. This intricate network is composed of three main types of protein filaments: microtubules, microfilaments, and intermediate filaments. Each type has a unique structure and function, contributing to the overall organization and dynamism of the cytoskeleton. Let's delve deeper into the world of these protein polymers, exploring their individual roles in maintaining the cell's structural integrity and facilitating its remarkable feats of cellular choreography. Microtubules, the largest of the cytoskeletal filaments, are hollow tubes composed of tubulin protein subunits. These dynamic structures are constantly growing and shrinking, assembling and disassembling as needed. They radiate outwards from the centrosome, a region near the nucleus, towards the cell periphery. Microtubules serve as intracellular highways, guiding the movement of organelles and vesicles. Motor proteins such as kinesin and dynin attach to these cargoes and walk along the microtubule tracks, transporting their precious cargo to their designated destinations. Beyond their role in intracellular transport, microtubules are essential for cell division. They form the mitotic spindle, a structure that separates chromosomes during cell division, ensuring that each daughter cell receives a complete set of genetic instructions. Microtubules also form the structural core of cilia and flagella, whip-like appendages that protrude from the cell surface. These structures enable cell motility, allowing cells to swim through liquid environments or move fluids past the cell surface. Section 3. Microfilaments, shaping the cellular landscape. Microfilaments, the thinnest of the cytoskeletal filaments, are composed of actin protein subunits. These two-stranded helical polymers form a dense network beneath the plasma membrane, providing mechanical support and contributing to the cell's shape. Microfilaments play a crucial role in cell motility, particularly in processes like amoeboid movement, where the cell crawls along a surface by extending and retracting pseudopodia. These temporary projections of the cytoplasm are driven by the assembly and disassembly of actin filaments. In muscle cells, actin filaments interact with myosin filaments to generate the contractile force needed for muscle contraction. This intricate interplay of protein filaments allows for the coordinated movements that power our every action. Microfilaments also contribute to cytokinesis, the final stage of cell division, where the cytoplasm divides, pinching off to form two daughter cells. The contractile ring, composed of actin and myosin filaments, constricts the cell membrane, eventually separating the two daughter cells. Section 1. The cell's vault, safeguarding the blueprint of life. Deep within the eukaryotic cell lies the nucleus, housing the cell's genetic material, DNA. Encased in a double membrane, the nucleus dictates cell activities and ensures genetic inheritance. Nuclear pores regulate molecule passage, importing proteins and exporting RNA. DNA is organized into chromosomes tightly coiled around histones. The nucleus safeguards and expresses genetic information, ensuring life's continuity. Section 2. The DNA symphony orchestrating life's instructions. Within the nucleus, the DNA molecule takes center stage, carrying the genetic instructions that dictate the cell's structure and function. This remarkable molecule, composed of two long strands twisted into a double helix, resembles a spiral staircase, with each step representing a nucleotide base. These bases, adenine, A, thymine, T, guanine, G, and cytosine, C, pair up in a specific manner, A with T and G with C, forming the rungs of the DNA ladder. This base pairing is crucial for DNA replication, ensuring that the genetic information is faithfully copied every time a cell divides. The sequence of these bases along the DNA strand forms the genetic code, a language that dictates the production of proteins, the workhorses of the cell. Genes, specific segments of DNA, provide the instructions for building these proteins, which carry out a wide array of functions, from catalyzing metabolic reactions to transporting molecules across cell membranes. The process of DNA replication is a symphony of molecular precision, involving a cast of enzymes that unwind the double helix, separate the two strands, and assemble new complementary strands using the original strands as templates. This intricate dance ensures that each daughter cell receives an identical copy of the genetic blueprint, preserving the continuity of life. Section 3. RNA, the messenger and the builder. While DNA holds the master blueprint for life within the nucleus, it is RNA, a single-stranded nucleic acid, that acts as the intermediary, carrying the genetic instructions from the DNA to the ribosomes, where proteins are synthesized. This flow of genetic information from DNA to RNA to protein is known as the central dogma of molecular biology. There are three main types of RNA involved in protein synthesis: messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. mRNA carries the genetic code from the DNA in the nucleus to the ribosomes in the cytoplasm. tRNA acts as a molecular interpreter, translating the mRNA code into a sequence of amino acids, the building blocks of proteins. RNA, a structural component of ribosomes, helps to assemble the amino acids into polypeptide chains. Our exploration of the eukaryotic cell now leads us to the endoplasmic reticulum, or ER, a vast network of interconnected membrane-bound sacs and tubules. Imagine a sprawling labyrinthine factory within the cell, its interconnected chambers bustling with activity. This is the ER, essential for protein and lipid synthesis. The ER is continuous with the outer membrane of the nuclear envelope, forming an intricate network throughout the cell. Its convoluted structure provides a large surface area for its functions. This extensive network is not static. It constantly changes shape. There are two distinct regions of the ER, the rough ER and the smooth ER. The RER, studded with ribosomes, is the primary site of protein synthesis. The SER, lacking ribosomes, plays a crucial role in lipid synthesis and detoxification. Together they form an integrated system essential for cellular processes. The smooth endoplasmic reticulum, SR, may appear simple, but it serves as a multifaceted workshop within the cell. It plays a crucial role in lipid synthesis, carbohydrate metabolism, detoxification, and calcium storage. Lipid synthesis includes cholesterol, phospholipids, and steroids. This is vital in liver, testes, and ovaries. The SCR also breaks down glycogen, releasing glucose. It detoxifies harmful substances, making them easier to excrete. Our journey through the eukaryotic cell now brings us to the Golgi apparatus, named after Italian scientist Camillo Golgi. Imagine a bustling postal service within the cell, sorting, packaging, and dispatching molecular mail. The Golgi apparatus consists of flattened membrane-bound sacs or cisternae stacked like pancakes. It receives newly synthesized proteins and lipids from the endoplasmic reticulum. These molecules arrive at the CIS face of the Golgi. As they journey through the Golgi stack, they undergo modifications and sorting. Enzymes modify the proteins and lipids, adding or removing sugar molecules. These modifications act as address labels, directing molecules to specific locations. The Golgi apparatus is dynamic, adjusting its activity to meet the cell's needs. As molecules traverse the Golgi apparatus, they are sorted and packaged into vesicles. These vesicles act as cellular delivery trucks. One major route leads to lysosomes, breaking down waste. Another route leads to the plasma membrane, releasing contents outside the cell. The Golgi is the cell's central sorting and shipping hub. Our exploration of the eukaryotic cell's intricate transportation system now leads us to endosomes, a dynamic network of membrane-bound compartments that act as cellular sorting stations. Imagine a busy package delivery depot within the cell, where incoming parcels are received, sorted, and dispatched to their appropriate destinations. That's the role of endosomes, which receive vesicles budding from the plasma membrane, capturing their cargo, and directing it to various intracellular compartments. These vesicles, formed during endocytosis, engulf extracellular material, bringing it into the cell enclosed within a membrane-bound sac. Early endosomes located near the cell periphery serve as the initial receiving point for endocytosed vesicles. As they mature, they transition into late endosomes, gradually acquiring different protein compositions and moving deeper into the cell's interior. The endosomal system plays a crucial role in maintaining cellular homeostasis by regulating the uptake and degradation of various molecules. It acts as a gatekeeper, controlling the entry of essential nutrients and signaling molecules while also eliminating harmful substances and cellular waste. Within the endosomal network, a complex interplay of sorting and trafficking pathways determines the fate of internalized molecules. These pathways, resembling a network of intracellular highways, direct molecules to their final destinations, whether for recycling back to the plasma membrane, degradation in lysosomes, or transport to other organelles. One major pathway involves the recycling of receptors and membrane components back to the cell surface. After their cargo is released, receptors embedded in the endosomal membrane can be sorted into recycling endosomes. These endosomes bud off vesicles that return the receptors to the plasma membrane, ensuring a constant supply for further rounds of endocytosis. Another crucial pathway leads to the lysosomes, the cell's digestive centers. Late endosomes containing molecules destined for degradation fuse with lysosomes, delivering their contents to these acidic compartments. Lysosomes are filled with hydrolytic enzymes that break down proteins, lipids, carbohydrates, and nucleic acids, effectively dismantling cellular waste and debris. The endosomal system also plays a role in transcytosis, the transport of molecules across a cell layer. In this process, molecules are internalized at one side of the cell, transported across the cytoplasm within endosomes, and then released at the opposite side. This mechanism is particularly important in epithelial cells, which form barriers between different tissues. Our journey into the eukaryotic cell now takes us to the lysosomes, small membrane-bound organelles that serve as the cell's recycling centers. Imagine these organelles as the cell's waste disposal and recycling plants, breaking down cellular debris and worn-out organelles. Lysosomes are spherical sacs filled with powerful hydrolytic enzymes, capable of breaking down a wide range of biological molecules. These enzymes operate best in the acidic environment maintained within the lysosome. Lysosomes play a vital role in maintaining cellular health by degrading waste materials and recycling valuable molecules. Malfunctions in lysosomal function can lead to lysosomal storage diseases. These diseases are characterized by the accumulation of undigested materials within lysosomes. When lysosomal enzymes are absent or defective, specific molecules cannot be broken down properly. This accumulation can disrupt cellular processes and lead to cell death. Tay-Sachs disease is caused by a deficiency in the enzyme hexosaminidase A. This enzyme breaks down GM2 ganglioside, abundant in nerve cells. In Tay-Sachs, the absence of this enzyme leads to GM2 ganglioside accumulation, damaging nerve cells. Lysosomal storage diseases underscore the importance of lysosomes in cellular health. Our exploration of the eukaryotic cell now takes us to the vacuoles, dynamic storage compartments found within the cytoplasm. Imagine these organelles as the cell's network of warehouses and storage lockers. Vacuoles are particularly prominent in plant cells, where a large central vacuole often occupies a significant portion of the cell's volume. In animal cells, vacuoles are typically smaller and more numerous, serving a variety of functions. Vacuoles can store a wide range of substances, reflecting their diverse roles in cellular function. Animal cell vacuoles exhibit a remarkable diversity in size, shape, and function, reflecting their involvement in a wide range of cellular processes. These dynamic compartments play essential roles in maintaining cellular homeostasis, acting as storage depots, waste disposal units, and regulators of cell volume and pH. One crucial function of animal cell vacuoles is their involvement in endocytosis and exocytosis. As we delve into plant cells, we encounter the vacuole, an organelle of remarkable size and versatility. Plant cells typically possess a single large central vacuole that can occupy up to 90% of the cell's volume. Encased within the tonoplast, it maintains structural integrity, growth, and health of plant cells. The tonoplast regulates molecule movement between the vacuole and cytoplasm. This regulation maintains the vacuole's distinct internal environment. The vacuole stores water, ions, sugars, pigments, enzymes, and waste products. It plays a fundamental role in plant growth and development. By absorbing water, the vacuole exerts pressure against the cell wall. This turgor pressure provides structural support, enabling plants to maintain their posture. One of the most critical functions of the plant cell vacuole is maintaining turgor pressure, the force exerted by the vacuole's contents against the cell wall. This pressure is essential for structural integrity, enabling plants to stand upright and resist wilting. Turgor pressure is maintained by water moving into the vacuole and the cell walls counteracting pressure. The vacuole's solutes create an osmotic gradient, drawing water in. As water flows in, the vacuole expands, pushing against the cell wall. The cell wall provides support and limits vacuole expansion. This balance prevents the cell from bursting under osmotic stress. Turgor pressure is vital for plant growth, development, and overall health. Our exploration of the eukaryotic cell now takes us to the peroxisomes, small, membrane-bound organelles that function as metabolic powerhouses. They carry out essential reactions in lipid metabolism and detoxification. Enzymes in peroxisomes generate and degrade hydrogen peroxide, neutralizing its harmful effects. They also break down very long-chain fatty acids through beta-oxidation. Additionally, peroxisomes detoxify harmful substances like alcohol and drugs. In the plant kingdom, glyoxysomes play a vital role in converting stored fats into energy for the developing seedling. These organelles provide a critical metabolic pathway for seedling growth before photosynthesis can commence. During germination, the embryo relies on stored fats and oils to fuel its growth. Glyoxysomes catalyze the breakdown of fats into sugars, providing energy for roots, shoots, and leaves. The glucose produced fuels growth until the seedling can photosynthesize. Once photosynthetically active, glyoxysomes decrease as the plant relies on sunlight for energy. Our exploration of the eukaryotic cell now takes us to the mitochondria, the powerhouses of the cell. These organelles convert nutrients into usable energy in the form of ATP. Mitochondria have double membranes with inner folds called cristae. The inner membranes cristae play a crucial role in ATP synthesis. The inner mitochondrial membrane, with its intricate folds called cristae, is the stage for the electron transport chain. Protein complexes generate energy to produce ATP. This process harnesses energy from glucose breakdown to create a proton gradient, driving ATP synthesis. Electrons are passed along protein complexes, releasing energy. This energy pumps protons across the membrane, creating a gradient. ATP synthase harnesses this gradient to produce ATP. Our exploration of eukaryotic cells now brings us to the chloroplasts, the remarkable organelles responsible for photosynthesis. They convert light energy into chemical energy. These green lens-shaped organelles harness sunlight to sustain the entire biosphere. Chloroplasts have a double membrane and a fluid-filled space called the stroma. Within the stroma, thylakoids form stacks called grana containing pigments like chlorophyll. Thylakoid membranes house protein complexes for light-dependent reactions. The stroma is crucial for the Calvin cycle, fixing carbon dioxide into sugars. The symphony of light, capturing energy from the sun. Within the thylakoid membranes, a remarkable molecular dance unfolds, capturing sunlight and converting it into chemical energy. The process begins when photons of light strike chlorophyll molecules. Excited electrons are passed along a chain of protein complexes, pumping protons across the membrane. This gradient is harnessed by ATP synthase to synthesize ATP. This intricate interplay converts light energy into chemical energy, fueling life on Earth. Beyond the membrane organelles without boundaries, our journey through the eukaryotic cell reveals membrane-bound organelles with distinct functions. But let's explore unbounded membrane organelles, essential yet often overlooked. Ribosomes, for instance, synthesize proteins without a membrane. The cytoskeleton provides structural support and aids in cell movement. These structures highlight the cell's adaptability and efficiency. Even without membranes, they play crucial roles in cellular life. The cycle of life. Cell division and renewal. Cell division ensures growth, repair, and continuity of life. It's a choreographed dance, duplicating and dividing cells. The cell cycle ensures accurate DNA replication and distribution. Each new cell gets a complete set of chromosomes. The cell cycle has interphase and the mitotic phase. Interphase is for growth and preparation. The mitotic phase is the actual cell division. Mitosis is followed by cytokinesis, forming two daughter cells. Mitosis, a symphony of duplication and division. Mitosis is a remarkable feat of cellular choreography, ensuring accurate replication and distribution of chromosomes. This intricate dance is essential for growth, repair, and asexual reproduction. Prophase marks the beginning of mitosis. Chromosomes condense and become visible as distinct structures. The nuclear envelope breaks down, releasing chromosomes into the cytoplasm. The mitotic spindle begins to form, serving as scaffolding for chromosome movement. A microscopic symphony of life. Our journey into the eukaryotic cell reveals astonishing complexity and elegance. From the cytoskeleton to mitochondria and chloroplasts, we've witnessed a symphony of structure and function. Like a bustling metropolis, the eukaryotic cell's specialized compartments maintain its vibrancy. The nucleus orchestrates activities directing protein synthesis. The endoplasmic reticulum and Golgi apparatus manage production and dispatch. Lysosomes recycle waste, while vacuoles store nutrients. Mitochondria generate energy, and chloroplasts capture light. These processes highlight the efficiency and adaptability of eukaryotic cells, underscoring the principles that govern all living organisms.